noteworthy individual in the history of neuroscience is Camillo Golgi, who in the late 19th century developed a staining procedure for observing individual neurons in the brain. One thing about the brain is that it is, from one perspective, a gnarly mess of neurons going in all kinds of different directions, meaning they're axons and dendrites. And so they're all interwoven in a way that makes it basically impossible to just look at a slice of brain and see an individual neuron, because it is being crossed over by so many other axons and dendrites, and it itself has so many dendrites and axons going out in different directions that it's hard to tell where the neuron ends and begins, and even where it is in between. So Golgi applied a particular kind of stain. It's become known as the Golgi stain. And he applied it to tissue and found that only 1 in every 10,000 neurons would absorb this dark dye. As such, that 1 in 10,000 neurons that absorbs the dye will pop out from the background, as we can see on the image, or in the image on the right. It has absorbed the stain, whereas all the other neurons with their dendrites and axons that are running to and from did not absorb the dye. So our neuron of choice, as it were, jumps out from the background, and we can see its cell body there in the center. We can see the axon running from the center of the image up to the top. We can see the dendrites on the opposite end of the neuron. And Golgi actually won the Nobel Prize for this invention, or I suppose it's more of a discovery. He won that Nobel Prize in 1906 for, again, what we call Golgi stain. Now, even though he did Nobel Prize winning work in terms of how to view the individual neuron, and this is, by the way, this is under a microscope, I should say, because his neurons are microscopic in size. Despite his great achievements, one thing that was curious is he seemed to stick to the idea that the fibers, namely axons and dendrites and the cell bodies, were essentially just water pipes carrying information around the nervous system. So he hasn't even fully taken on the idea that nerves are conducting electricity. He's still kind of stuck on the water pipes issue, so kind of surprising, but not to take anything away from the man. Had some great achievements. While Camillo Golgi discovered the staining procedure for which he won the Nobel Prize, he also shared that Nobel Prize with someone called Santiago Ramon y Cajal, a Spanish neuroscientist, pathologist, physiologist, who improved slightly upon Golgi's staining procedure and was the first person to systematically look at these different neurons in the brain under the microscope and make detailed drawings of them. Cajal was always an artist of some sort, though his father discouraged him from pursuing art. He nonetheless managed to bring his art into his neuroscience career in the form of detailed drawings of the neurons that he looked at through the microscope. And again, he's one of the first people to be looking at these neurons through the microscope. These drawings he made are still used today, oftentimes in textbooks and stuff, as we can see over here on the right, just because you don't get any better depictions of these neurons than the drawings that Cajal made. Cajal's observations led him, at least indirectly, to the neuron doctrine, which is the idea that the neuron is the functional unit of the nervous system. The neuron doctrine says that neurons are individual units, each autonomous, so to speak, on their own. By contrast, Camillo Golgi, who thought these were water pipes, thought they were kind of continuous fibers that ran, well, continuously, that they were not disconnected. Cajal posited that they were not actually connected, that individual neurons were separate from each other. And this is true. As you will know from intro neuroscience, there is a cleft in between the axon of one neuron and the dendrites of another neuron, the synaptic cleft. They don't actually touch each other. Okay. Now, Cahal had microscopes and the Golgi staining procedure at his disposal, but these were not high-resolution microscopes, at least not in terms of being able to see a synaptic cleft. Cahal was not able with his microscopes to see a synaptic cleft. Nonetheless, and I'm not sure why, but he believed there was a cleft there, a space. And that space meant that neurons are individual functional units. And if neurons are individual functional units, then this gives rise to the neuron doctrine. Having turned from the 19th to the 20th century, we can now speak of Ross Harrison and his work in support of the neuron doctrine, the idea that the neuron is the functional unit of the nervous system. And I say his work, but generally all work is kind of confirmed the neuron doctrine. But specifically with Ross Harrison, we have the idea that dendrites and axons grow out of the cell body. This was not obvious. Here they are, thanks to the Golgi staining procedure, they're able to see individual neurons, and they can see that individual neurons have a cell body, have a primary axon, and a dendritic tree. But it's not clear where the axons and the dendrites come from. Do they grow out of the cell body, or are they manufactured by some kind of third party? But not only did Ross Harrison help demonstrate that dendrites and axons grow out of the cell body, but he also showed that axons are guided by growth cones. Growth cones are something that occur in the development of the brain, where radioglial cells will develop these growth cones that guide neurons where to go in this developing brain. So as regards the growth of the nerve fiber out of the brain, there were three theories about the origin of the developing nerve fiber. One was the cell chain theory, that the nerve fiber, this is the axon or the dendrite, the axon is formed by cells that form the nerve sheath. So for instance, you'll recall from intro neuroscience that oligodendrocytes produce these fat arms that wrap themselves around axons and give them insulation. Well, one theory at the time was that what these myelin sheaths were doing was not only insulating the axon, but actually creating the axon. Maybe it's not that the axon grew out of the cell body, but that these oligodendrocytes lined themselves up and produced an axon and attached it to the cell body. That's the cell chain theory. The plasmoderm theory was another one that this suggested the fiber is formed by preformed protoplasmic bridges, that there are these basically kind of scaffoldings in the space around neurons, and that this scaffolding, these protoplasmic bridges, form the axon. So one is oligodendrocytes are forming the axon, the cell chain theory. The second one is the plasmoderm theory that says that these protoplasmic bridges are forming the axon. And then there's the correct theory, as it turns out, the outgrowth theory. This is the theory that the fiber is the product of the nerve cell itself. Rather than being created by oligodendrocytes or by preformed protoplasmic bridges, the axon under the outgrowth theory is believed to have grown out of the axon. And this is the correct theory, and we know this thanks in part to Ross Harrison's work, in which he was one of the first people to grow neurons in situ, without nerve sheaths or protoplasmic bridges, leaving the outgrowth theory the only available alternative. So he took neural tissue, put it in a petri dish, and took away the oligodendrocytes and protoplasmic bridges, and yet an axon still formed. This left only the third theory being possibly correct, that axon had grown out of the cell body. A little bit after, not so much after Ross Harrison, but a little bit younger than Ross Harrison was Sanford Pele, who was the first person to use an electron microscope to look at the synapses of axons with dendrites. As we said, Ramon Cajal came up with his neuron doctrine, stating that the neuron was the functional unit of the nervous system, and he believed that it's a functional unit into itself because it is detached from all other neurons. There's a space between them. And Sanford Pele was the first person to observe that space, that synaptic cleft, using the electron microscope. Another important person in the history of neuroscience, particularly the early 20th century, was a person by the name of Margaret Kennard, who pioneered neuroplasticity research. She was the person that came up with what is now called the Kennard principle. Younger brains recruit from lesions more efficiently than older ones. This is something that I think many people are familiar with, the idea that, for instance, if you're going to have to suffer a traumatic brain injury, it's better to suffer it as a young child than it is as an adult, because the young child's brain is more plastic. It has more neuroplasticity and therefore can heal better, whereas the older brain is less neuroplastic and therefore does not heal as well. And so this well understood principle, one that's still important today, has been known as the Kennard, probably better pronunciation is Kennard principle. We'll end our discussion of the history of neuroscience by talking about the current status of the field today and what the different subdivisions are. We have skipped over much of the 20th and early 21st century, mostly because what we discussed this semester or during this course will be discoveries from the last 100 years. So rather than go over that stuff now, I will just kind of mention what neuroscience looks like today. And we can think of neuroscience in terms of levels of analysis. And this is relevant to a reductionist approach. Reductionist approach being the idea that the psychology of it all can be reduced to neural activity. And in terms of levels of analysis, we have the smallest level being the molecular neuroscience level. This is looking at the myriad molecules that play different roles in nervous system function. Okay, so you could be looking at things like neurotransmitters or various other molecules that are in the brain and are relevant to brain function. Now, instead of looking at molecules, you may be interested in a slightly larger scale, a larger level of analysis or a higher level of analysis, looking at individual neurons or cells, cellular neuroscience. In this case, you're looking at how do these molecules interact to create a functioning cell. So your unit of analysis is not the molecules themselves, although you'll pay attention to molecules coming in and out of the cell because that's relevant to the cell's function, but your primary interest is how does the neuron work. Now, understanding the molecules that are involved and understanding how the neurons themselves work is extremely useful in many respects to understanding psychological phenomena. But many of the higher level psychological functions that psychologists tend to be interested in are better seen at the systems neuroscience level. This is looking at how do molecules and cells interact on a large scale to form networks or systems. So, for example, the visual system. The visual system, I honestly am not sure exactly how many neurons there are in the in the visual system, but at least hundreds of millions, perhaps up into the billions. but that's a lot of neurons. And so if you're just looking at a single neuron as a cellular neuroscientist, you're looking at a single neuron that is one of a billion neurons in a network, then you're seeing a very small part of the story. Now, having said that, that's still a very useful part of the story, but to really understand visual processing, you really have to understand how these neurons work together to form the process of vision. Now, as a molecular, cellular, or systems neuroscientist, you can fall into one of two kinds of general types of neuroscience: behavioral neuroscience or cognitive neuroscience. Behavioral neuroscientists are usually those people who are doing invasive work. They're often doing systems level analysis, but they can certainly do cellular or molecular. And they generally are using non-human animals as their subjects because they're usually doing brain surgery of some sort, or maybe creating lesions, or what have you. So invasive work is usually done by people calling themselves behavioral neuroscientists. On the other hand, non-invasive work using things like brain imaging, functional magnetic resonance imaging, electroencephalography, magnetoencephalography, positron emission tomography, these different non-invasive brain imaging techniques are typically used by people calling themselves cognitive neuroscientists. As such, behavioral neuroscientists are a little more likely to be found in biology departments, although plenty of psychology departments have them. And as such, cognitive neuroscientists are more likely to be found in psychology departments, although some biology departments will have cognitive neuroscientists.